Okay, this is part 2 of the lesson. We're going to go over some ratios and then incomplete and codominance. So, after the slideshow today, you should be able to explain the effect of codominant alleles on the phenotype and explain the effect of incomplete dominance on the phenotype, and then list the ratios for various monohybrid and dihybrid crosses. The ratios, it would be nice if you're able to memorize this stuff, but you can always figure it out by creating the Punnett square. So this is just kind of an added benefit for some of you who have a natural aptitude for this stuff. So pause the video and with your table discuss what incomplete dominance and codominance might mean. Alright, thank you for talking about this. Now, let's talk about incomplete dominance. The definition. This is the definition out of your book, so go ahead and write it down. But really the simple version is that incomplete dominance is when two traits mix. The example is the red color flower and the white color flower. They're not completely dominant one over the other, so you end up with pink flowers. Generally we'll pick one as the dominant trait. We'll say that red is dominant, and we'll say that white is the little or the lowercase letter, but we know that it's not completely recessive or completely dominant, and we get a mix of the two, these pink flowers. So let's take a look at a cross for an incomplete dominance. Feel free to pause the video while the kids copy down any notes. For flower color, red and white show incomplete dominance. So we're going to cross a homozygous recessive white flower with a homozygous dominant red flower. Now, when you get a heterozygous individual, you get the phenotype pink instead of red or white. So we're going to bring our capital R's over from our red plant, and then we're going to bring our little r's down from our white plant, even though it's not really recessive. Now let's list the genotypes and phenotypes. So let's do a different cross. We've got one parent here that we're going to put at the top. We're going to put another heterozygous individual here. So we've got pink plants, right? They're heterozygous, so we've got pink plants. Let's do the cross. And we have red plants. So our ratio is 1 to 3. White plants, the ratio is 1 to 3. And pink plants, the ratio is 1 to 1 or 50%. So the phenotypes, instead of just having two phenotypes like we've had in the past, the phenotypic ratios are actually going to match the genotypic ratios. So red ones is the same as our capital R's up here. White is the same as well. And then our pink ones, 50%. Feel free to pause or play this part again if you need to. Let's go on to codominance. Here is when you actually see both alleles. This may be kind of hard to imagine, so here, let's talk about an example. Roan is a type of coloring in animals most often associated with horses, and it happens when both colors of hair are fully expressed in the animal. So from a distance, the animal looks like it has a different color hair. Alright, that still may be confusing, so here's a picture. Here is a roan horse. From far away, the horse may look a pretty gray color, but it's actually there are lots of black hairs and white hairs all growing side by side in here, and it gives the horse a gray appearance. So in codominance, the white hair is not dominant over the black hairs, but neither do they mix. You end up with white hair and black hair. This is called a strawberry roan, where you get a blonde or wheat color with white, and it creates its own coloring. This is actually what's going on with spots on dogs as well. The different colored spots, one's not dominant over the other, so you end up with a white dog with black spots or something similar. So let's take a look. We've got a brown horse and a white horse, and the hair color is co-dominant. So in their offspring, both of those colors will be expressed if they carry those traits. Now in this case, we can't do a little r for the recessive, so we're going to put an apostrophe next to one of the colors. So we're going to use RR for brown. We're going to use an R with an apostrophe for the white. A roan horse would have one R without an apostrophe for brown and one R with an apostrophe for white. And both of those hair colors show up on the horse. So let's cross one that is white and brown. So there are the genes for the brown hair. Now let's bring down the ones with the apostrophe that indicate our white coloring. Sorry, these are on top of each other. But they all look like this. So all of the offspring will be roan because they have a gene for brown hair and a gene for white hair. So they end up with both. So let's do it this way. Let's do two heterozygous individuals. Oh, and there's a little one. It's supposed to look like this. Now, let's do our genotypes and phenotypes. And just like in incomplete dominance, the genotypes and the phenotypes are going to have the same ratio. So the brown horse, the two capital R's with no apostrophes, is 1:3. The white horse, with two apostrophes, is also 1:3. And then the mix, the roan horse, that has one of each allele, is 1:1. So we have 1/3 brown, 1/3 white, and a 1:1 ratio of roan. Pretty simple. There aren't a lot of questions about these on the test, but you do need to understand what they are. You also need to be familiar with what this means, and it means co-dominance. It will take some effort on your part to distinguish that from incomplete, where we still use a capital letter and a lowercase letter. So some ratios. When you cross a homozygous recessive and a homozygous dominant for monohybrid crosses, the genotype will always be 100% heterozygous. Hopefully that's a little familiar by now. The phenotype will also be 100% dominant because you have the dominant gene. When you cross a heterozygous individual with another heterozygous individual, you get several different genotypes. The ratio here is considered 1:1. One dominant, two heterozygous, and one recessive. The phenotype for this, however, is not the same. It's that 3:1 that we're used to seeing, where you have 3 that show the dominant trait and 1 that shows the recessive trait. For a heterozygous crossed with either a homozygous recessive or a homozygous dominant, you get a 1:1 ratio. 50% are heterozygous and 50% are homozygous. And the phenotype is 1:1 as well. And actually it looks like we're crossing this with a homozygous recessive. So the ratio there is 1:1. If it was homozygous dominant, it would look like this up here. Please feel free to rewind and play this part again if I went too fast. Dihybrid. You won't see any of these ratios, but I know some of you can handle it, so here's a little extension. If you cross a homozygous dominant and a homozygous recessive for both traits, we know that 100% of the offspring will be heterozygous for both traits, meaning the phenotype, 100% of them will be dominant for both traits. Let's say you cross a heterozygous individual with another heterozygous individual for 2 traits. This is the phenotype. We're not even going to talk about the genotype because that's going to get a little messy. But the phenotypes, it's a 9-3-3-1. And what that means here is 9 will show the dominant trait for both, 3 will show the dominant and a recessive, and 3 will show the recessive and a dominant. and 1 will show all recessive traits. This is the only ratio that you're likely to see, this 9-3-3-1. If you see this 9-3-3-1, just know that it's a dihybrid cross because there are 4 numbers in our ratio, and that it's a heterozygous for both traits crossed with a heterozygous for both traits. Both of these screencasts are on my YouTube channel. You can get to it from my website. There's now, we'll talk about, quickly, a test cross. When you want to know the genotype of an individual. Obviously, if it's showing the recessive traits, you know that it's homozygous recessive, but if it's showing the dominant trait, you don't know if it's homozygous dominant or whether it's heterozygous. So you would cross this individual with another individual. Which one would you pick? Hopefully you picked the homozygous recessive. The offspring would then tell you whether or not the original parent is homozygous dominant or heterozygous. So now there is a worksheet on incomplete and co-dominance. We'll go over this before the test on Monday, but again, there are only 1 or 2 questions about incomplete and co-dominance on the test, and there are no Punnett squares for those. Please feel free to play this again or watch it this weekend, and email me with your questions because I get my work email at home. It's GaletteP@rcschools.net. Have a good weekend, guys.